immunodeplete here in the right lane of this western blot, immunodeplete the A beta within the brain extract using antibodies to A beta on beads, what we see is that the seeding effect of the extract compared to the normal extract is eliminated. So this was pretty strong evidence that it's A beta, but one piece of the puzzle remained elusive. If A beta is the seed, then you should be able to make synthetic A beta aggregates and seed the process in the brain with these, right? Well, the prion people had been dealing with this issue for many years unsuccessfully. It just seemed that prions that were made synthetically or recombinantly didn't work the same way that prions made in the brain. In fact, it's one of the main objections that the opponents of the prion theory have had to prion disease, since they have shown that it's possible. So what we did was to prepare A beta extracts, aggregate them, basically then do everything we did with the brain extracts. And this concentration of synthetic A beta is about the concentration that was in the Alzheimer brain extracts. And then after our usual 5 months, we looked to see what happened. And what happened, disappointingly, but not surprisingly, was nothing. We just didn't see anything. We did many variations on this, and there was occasionally a hint that maybe some seeding effect was taking place, but it was not convincing. This year, however, the Prusner lab has done the same sort of study, but they have used a much higher concentration of the A beta, and more importantly, I think, they waited much longer. So if you wait longer, you do see with synthetic A beta evidence that A beta itself is seeding this process in the brain. It's not nearly as effective as A beta that's aggregated within the brain, but it does work. So it does appear that A beta is able to induce this seeding cascade. So what we think is happening in Alzheimer's disease, Alzheimer's isn't infectious in the same sense that prion disease is. What we think is happening is that there's endogenous misfolding of A beta, and this endogenous seed then can ramify throughout the brain and cause Alzheimer's disease as we know it. So it misfolds, does everything basically the way that the prions do, and eventually the cascade leads to the characteristics of Alzheimer's disease, including tangles, loss of neurons, and dementia.
Larry?
Yes?
Is there any possibility that there's something else stuck to the endogenous A beta that's facilitating this process that you just can't make with the synthetic?
Yeah, there is. ApoE, for example, is a risk factor for Alzheimer's disease and is related to A beta. There are metals that are associated with A beta. So some of the control experiments that we ran was to mix the A beta with control brain extract, with metals, with ApoE. None of these things seem to make any difference. Now, it could be that there's a chaperone. What we think is happening is that there may be a chaperone in the living brain that somehow controls the architecture of the misfolded protein, and we think that that architecture is critical to the pathogenicity of the protein. So how do these seeds spread through the brain? Pathologists have noted for many years that Alzheimer's and actually many other neurodegenerative diseases seem to progress from one part of the brain to the other. And the seeding paradigm, we think, offers a possible explanation for how this is happening. So what we've done is when we inject the Alzheimer brain extract into the dorsal hippocampus of a mouse, and you can see these are actually taken from the same tissue section. What we have noticed in many cases is down in the entorhinal cortex, there is an isolated patch of A beta in many of the mice that you don't see anywhere else in the brain at this time point. You don't see it anywhere else in the cortex. And as you know, the entorhinal cortex and the hippocampus are big communication partners. There are lots of axonal connections between them. And this suggests that maybe axonal transport and not just diffusion might be a directed means of the trafficking of the seeds from one region to another. Another thing that we asked was, well, is it possible that these seeds can get into the brain from the periphery? So Alzheimer brain extracts or transgenic mouse brain extracts, which work just as well as long as the transgenic mice have lots of plaques in the brain, inject them into the peritoneal cavity and then look sometime later to see what happens in the brain. In this case, there was a longer incubation period. And what we see is that in this particular model, here at higher magnification, you can see that there's A beta deposition in the blood vessels, what's called cerebral amyloid angiopathy in these mice. And it is considerably increased in animals that receive the A beta rich brain extracts in peritoneum compared to animals that receive a control brain extract. So this indicated that the seeds can move from the periphery to the brain. But it wasn't clear exactly how they were doing this, because when we injected, and this was done in collaboration with Matthias Yerkes' lab in Tübingen, when we injected the seeds directly into the bloodstream, we didn't get this same effect. What we did see, what Matthias's lab has seen, and what our lab has seen, is that, and this was Amaryllis Sintrom's work, is that there are occasional macrophages in the blood that take up the material that is injected into the peritoneal cavity. And the question then was, well, is it possible that these macrophages are transporting the seeds into the brain? So to get to that question, we have to ask first, well, do macrophages traffic from the peritoneal cavity to the brain? So what Amaryllis has done is to take fluorescent nanobeads, just a very small number, and inject those into the peritoneal cavity of mice. And what happens then is that these are taken up, every one of these at low magnification is a macrophage. It's just full of these fluorescent nanobeads. And then the question is, well, do these get into the brain? In other words, do the macrophages traffic these to the brain? And in fact, we do see occasional macrophages that are full of these fluorescent beads within the brain. The number is very small, and we've only looked at very limited time periods for this. But there's no question that there are some in the brain. So then the next question is, well, does this eventually lead to the formation of A beta deposits in the brain? And that's what Amaryllis is working on right now by injecting the Alzheimer brain extract into the peritoneal cavity, looking to see whether it's in the macrophages, then taking those macrophages, isolating the macrophages from one mouse, and infusing the macrophages into another mouse of the same strain, and looking to see where they go and whether they will induce this process. And we don't have the answer to that quite yet. So what are the implications of this concept? How might this lead to some way of dealing with, of treating these sorts of diseases, which currently have no effective treatment whatsoever? Well, the first obvious thing we can do is to prevent the formation of the protein that's aggregating in the brain. And in the case of Alzheimer's disease, of course, it's, the protein is A beta. And if we block protein formation, then we can prevent it from eventually going on to cause the disease. Another option might be to block the misfolding and aggregation. These interact with cells and somehow kill the cells. If we can prevent that, that might help. If we can prevent the aggregates from breaking apart, that could limit their spread to other cells. And finally, there are ways that we may be able to promote the removal of these aggregates from the brain, perhaps, for example, by stimulating the heat shock response or stimulating transporters, which we know move A beta from one region to another. And in fact, these approaches are being, some of them at least, attempted with regard to Alzheimer's disease. There are attempts to develop enzyme inhibitors for the enzymes that cleave A beta from its precursor protein. That's one of the hottest areas of research in Alzheimer's disease. Aggregation inhibitors have been a little bit of a bust, and there are chemical reasons, I think, why that probably is. These are still highly hypothetical. One of the areas that's gotten the most attention in recent years is the idea that you can immunize patients against this protein and then allow the immune system to remove the protein from the brain. It works, but it hasn't been shown yet to slow the progression of Alzheimer's disease in any significant way. We think what's happening in that regard is that we simply are starting too late, that we need to treat people before they show up at the doctor's office with symptoms. And finally, just to point out that this isn't just prion disease, it isn't just Alzheimer's disease. There's a growing list of diseases that involve protein aggregation. And here in bold are those proteins that have now been shown to be transmissible, to be inducible in terms of their architecture in a prion-like fashion. So, and in fact, this is really just the tip of the iceberg. And this accounts for a very large number of the degenerative diseases of old age. If you include diseases, the amyloidoses of the liver, spleen, bone, and blood, there are all sorts of different diseases, cataracts, that involve protein aggregation. In fact, at my last count, the number is well over 50 or 60 different diseases that involve protein aggregation. And whether they all involve this prion-like mechanism of templated corruption of protein structure remains to be determined. But it is in aggregate, even though a lot of these diseases are relatively uncommon, in aggregate, it is a huge clinical problem. So in conclusion, I hope that I've convinced you that I think this is an important concept. I think it's a potentially unifying concept for many of the diseases of old age. It's worth pointing out that although I focus on A beta, tau has to be brought into the mix because tau is a very important component of Alzheimer's disease. And I also should mention that although you can't ignore the role of A beta in Alzheimer's disease, it's central to the disease process. As we get older, in these idiopathic forms of Alzheimer's disease, we really don't know what's causing A beta to start accumulating in the brain. So there are still some upstream factors that are legitimate therapeutic targets for Alzheimer's disease. But I think if you're going to consider the disease in its entirety and at its core, A beta must be considered. At the molecular level, these aggregates and prions have, I think, very important and very illuminating similarities. And this whole story sort of indicates that sometimes thinking outside the box, getting outside the box, leaving your comfortable home and going to the Wellcome Museum to see what's happening can be extremely valuable. And indeed, who would have imagined that we're studying prion disease, we're studying scrapie in the 1930s, that this was going to tell them something about Creutzfeldt-Jakob disease in the 1960s and possibly Alzheimer's disease today. It would have just been unthinkable. So sometimes these connections are there, and they just need to be ferreted out. Again, Alzheimer's disease is not infectious, and I don't want to imply that it is, but I think at the molecular level, it has very important commonalities with prion disease. And I think that in future studies, in fact, Anil and David Lynn are here. We just met this morning to discuss the possibility of trying to link together these changes in proteins that take place at the molecular level to what eventually happens in the disease state. I think this is where the disease is headed, and I think this is what we have to learn. We have to know why these different structures cause the proteins to behave very differently within the body. And again, I'd like to thank my lab members, and I'm happy to take some questions.
You mentioned that you guys tried these experiments where you inject the A beta to the peritoneum and then look for it in the brain. What about experiments where you inject it into a ganglion, like a foot ganglion that looks like the motor system?
Yeah, so the question is, what if we were to inject this into a ganglion or a neural structure, basically, and see what happens? We actually have been, one of the things Amaryllis has been doing is, if you inject a retrograde tracer into the peritoneal cavity, what happens in the brain? What would you all predict happens in the brain if you inject a retrograde tracer in the peritoneal cavity? You might want to guess. What would you see? Where would you see it?
You see it in the motor neurons in the brain.
That's right.
Yeah.
So the vagus nerve, of course, the wanderer is the motor nucleus in the brainstem that innervates much of the peritoneal cavity. And in fact, the dorsal motor nucleus of vagus is the one area where we see neurons that have retrogradely transported this material. We haven't ruled out the possibility that neurons may be transporting from periphery to the brain, but we never see pathology with regard to A beta in the dorsal motor nucleus of the vagus, and we don't see it in the circumventricular organs as well. So we really think that it's something else, that the pathology appears in the cortex and not where we see the neuronally activated seeding that might occur in the brainstem.
So in relation to the synthetic A beta, it seems to me that even though this sort of demonstrated proof of principle, it's still a mystery in terms of concentration, and I'm wondering if you could take synthetic A beta, put it into a brain that has disease, so be it congenital, how do you have it sort of in a membrane that only allows small molecular weight molecules to see if you can identify cofactors required for acceleration?
Yeah, there are lots of, I think, very important things that need to be done with the synthetic material. One of the questions about transport in general involves small structures like exosomes, for example, are thought perhaps to be involved with the cell-to-cell spread of some of these, and it may well differ from one situation to another. We really think that synthetic A beta, when it aggregates, is different from the A beta that aggregates in the brain. And one bit of evidence we have for this, and why I think it's a structural problem, it's a molecular structural problem, is that if you form fibrils of synthetic A beta, they will not bind certain compounds that fibrils formed in the living brain will, like Pittsburgh compound B is one of these. So there seem to be some differences. Also, the synthetic A beta fibrils are much more sensitive to proteinase K than the fibrils that are formed in the brain. We think that it's a structural thing, and some of the other Prusner studies indicate that it's not cofactors. They've also purified A beta from the brain, eliminated virtually everything else, and it seems to see just as well as the A beta that's unpurified.
So from a naive point of view, it seems to me that it's been difficult to determine the function of the native protein prion in APP. Is that a true observation, and why?
It's definitely a true observation. So the ATP, the long precursor protein, which is about 695 amino acids long, is thought to be involved in the connection between cells and maintaining cell adhesion in some way. And there was a guy speaking here just a couple of weeks ago, Orencio, who presented a fairly compelling case that the A beta is necessary for the formation of memories, for cognitive functioning. That it's released normally along with neuronal activity, and that this is necessary at physiological levels. I think these things must have a function. Nature usually doesn't release little fragments of proteins or do anything in this way unless it has some biological function. Why we haven't been able to figure out with certainty what it is, I don't know, but we haven't.
Well, I think the backup would be if APP knockout mice are viable.
APP knockout mice are viable. They do fine. I mean, there's some evidence that they may have some abnormalities, but by and large they do fine. And it's thought because there's an APLP, an APP-like protein that compensates for its presence. If you knock both of those out, then the mice are not viable. So APP itself isn't necessary. So hold on, I'm going to go to the back.
Do the mice that were seeded with the Alzheimer's tissue, do they show cognitive deficits similar to that seen in human patients?
Our original studies, we actually did water maze analyses of the mice, and we didn't see anything very obvious. But you notice, well, actually, you may not have been able to see this, but only one hippocampus was affected, and it's mainly a diffuse form of amyloid. We don't see any evidence that there's pronounced neuronal loss in these, and we didn't see any evidence of behavioral change. I think in order to do the right behavioral study, we need to inject a lot bilaterally and let it really accumulate in the brain and then do behavioral studies. And in fact, Matthias Yerkes' lab has done that. They have a mouse that doesn't start developing plaques until it's 15 months of age. If they inject them at 3 months of age bilaterally with a small amount of A beta, by 15 months the brain is full of A beta deposition. Those would be perfect for doing behavioral studies, but neither he nor we have done such a thing yet. In theory, it should be possible. And in fact, in theory, it should be possible to get to the question of this structure, to perhaps, by injecting the Alzheimer brain extract, create an Alzheimer-like conformation of the protein in the mice. We have weak evidence at this point that that might be happening, but it's very weak. But it should be possible.
Yeah, the one positive thing about that is the neuronal specificity of these disorders, right? I mean, they all have probably a common feature. The question is why, because they definitely when they start, they are quite specific in where they start, what particular neuron they hit, and then they spread. So is there what all the thought about that in that about the specificity, the neuronal specificity?
Yeah, well, neuronal specificity can result from all sorts of things. I mean, my feeling is that it's related to two main things. Neurons are very, very active cells, and the neurons don't turn over. In other words, they're pretty much always there. And so things that happen in neurons tend to accumulate. And I think in all of these diseases, and there's evidence for this in Alzheimer's, ALS, Parkinson's, all of these things, I think what happens is that there's a more or less stochastic misfolding of a protein in one part of the brain, one cell perhaps, that isn't taken care of by the cell. And where it begins is determined by the genetics and the energy state of that particular cell, that particular region, and then it spreads from that region throughout the nervous system. Take, for example, ALS, which is well known to be a motor disorder, but it can manifest first as something affecting the spinal cord, something affecting the brain stem, and then there's frontotemporal dementia, which is, from a molecular standpoint, a very similar disorder, but which involves completely different changes. I think the reason they're different is not molecular, but rather it's a result of where in the nervous system this process first begins. And that, where it begins, and one of the questions is, why are certain areas vulnerable? We don't know.
Given that you can transmit the amyloid pathology, by injecting the disease brain tissue, into naive animals, just like was done in the prion studies, then why isn't Alzheimer's infectious? The prion disease is like Scraper, so clearly infectious and spread between animals and the chronic wasting disease, but yet, presumably patients with severe Alzheimer's, they're shedding misfolded A beta, and so why isn't that transmissible between patients in the same way that Scrapie is?
Yeah, we think that the A beta Cs are much more fragile than prions are. Prions can be maintained, for example, with regard to Scrapie. You can take the sheep off a field that was infected with Scrapie for 15 years, put another batch of sheep on the field, they will develop Scrapie. So the Scrapie agent seems to be very resistant to destruction, and the A beta Cs are, we believe, more fragile. Now, there are groups looking at the question of whether it might be possible to transmit Alzheimer's disease in certain unusual ways. For example, one of the forms of Creutzfeldt-Jakob disease that has been one of the 700 known cases of human transmitted Creutzfeldt-Jakob disease was caused by the injection of human growth hormone. Growth hormone used to be extracted from pituitaries, from cadavers, and they would take a batch of 10,000, 15,000 pituitaries, grind them up, extract the growth hormone, and then inject it into kids, and it worked like a charm. I mean, they would grow, but then they discovered sometime later that a fraction of these kids developed Creutzfeldt-Jakob disease. The number is approximately 200 for that as well. And the question is, might this also be the case for Alzheimer's disease? And there is a group that's looking at that question right now with very long-term incubation. Is it possible that these folks might be? And the evidence so far is that they are not. So the good news is it doesn't look like Alzheimer's.
That's good. You had mentioned that the chronic wasting disease might be spread by nose-to-nose contact by the deer, so maybe it wouldn't be a good idea to have extensive nose-to-nose contact with severely Alzheimer's patients. Maybe should be avoided.
Is there any A beta showing up in the blood that we could use test to see how, before you have symptoms, whether or not you're.
Absolutely. So there is
Get there?
There is A beta in the blood. And in fact, one of the main tests for Alzheimer's disease involves, it is in the blood, but the closer you are to the brain, the better. So usually they measure it in the CSF. They measure A beta and tau in the CSF. What happens in the early stages of Alzheimer pathogenesis is the A beta levels drop and tau levels go up in the CSF. Dennis Selko says that this is absolutely the most sensitive way of diagnosing Alzheimer's disease using this fluid. And I showed this just to indicate this is a hypothetical sequence of events, but it shows that this decrease in A beta in the CSF is the very first thing that happens. And the very last thing that happens in this cascade is the development of dementia. It's only at this point that the patients go to the doctor and say, I'm not remembering things so well. So yes, it's there, it's measurable. I think it has to be done in the CSF to be meaningful because the blood, the variability is just too high. The thing about this is, and the reason I think it's important to start these therapies very early, is that this starts happening 20 to 25 years before the symptoms of cognitive decline really begin to manifest themselves. And at this point, when cognitive decline is down, the amount of A beta deposition in the brain has peaked. And that may be why a lot of the therapies haven't worked yet.
What do you think is going on with the cellular and the biological process of aging itself that is increasing the likelihood?
I think that's the big question. Why is aging the major risk factor for Alzheimer's disease and really every neurodegenerative disease? We don't know. My guess is, there's proteostasis, the idea that proteins are made, they're degraded, they're transported, all of these sorts of things. Proteostasis declines with age. We know that for a fact. And my guess is that it simply makes a more permissive environment for development and the sustenance of these pathogenic seeds.
Well, I kind of remember that you mentioned that why Troy's group observed the plaque after the injection, while your group did not observe this kinetic evidence because they waited for it a long time. So that reminds me of an interesting observation in the comparative anatomy. It seems that the humans are uniquely valuable in some baseline of human genetic. Then do you think that, we know that human longevity has a longer longevity compared to the cat. Do you think that the longer human longevity may involve?
Yeah. So if you all don't know it, I hope you do now that Alzheimer's disease is uniquely human. We've never seen it in any other species.
Right.
And the question is why? The question is it because humans live longer? We don't think so, and we don't think so. I mean, it's possible that simply the number of years is at play. But there are very rare cases of Alzheimer's that occur in the late 20s and 30s. And chimpanzees can live to be 60. And yet chimpanzees, even though they have all the molecular machinery for Alzheimer's, we have yet to see in any non-human primate the full manifestation of Alzheimer's disease, even though they can have a fair amount of A beta in the brain by the time they die. So everything can happen at a reduced time scale, but it doesn't lead to Alzheimer's. And I think that's a really, really important question that comparative studies can answer. Okay, well, it's after 1. Thank you very much.